Hello, everyone. Welcome back from lunch. I'd like to thank the organizers for having me here. It's been really great. It's my first time to SANS, and I'm having a SANS, having a great time so far. So, in the words of Monty Python, now for something completely different. So, what my lab works on is how neural circuits control adaptive behaviors. Specifically, we focus a lot on survival behaviors, such as the behaviors that promote acquiring food or avoiding threat, or perhaps the behaviors that allow you to survive multiple trips back to the hotel to fix your presentation 15 minutes before you present. But anyways, to illustrate what, I work in rodents, so to illustrate what rodent defensive responses to threat look like, which is what I'm going to be talking about today, I have this video that I lifted from YouTube. As scientists, we're not allowed to do experiments like this without ethical approval, but people on YouTube can do whatever they want.
You have got to be kidding me. That mouse is fast. Look at that. He's just like, come on.
So this is the well-characterized freezing response that you've probably seen measured in many, many papers. It's a rigid immobility, a complete cessation of bodily movements except for that required for respiration. And it's believed to have evolved to avoid detection by predators.
I think they go by motion.
So on the second pass there, you saw the mouse adopted a very different defensive strategy. That jumping and forward locomotor response is what's known as an escape response or a flight response. And I won't show you the end of the video because it does not end well for the mouse. But understanding, so there's a prominent psychological theory called threat imminence theory, which puts forth that different species have sets of defensive responses to threat, and the type of response that's chosen is dependent upon the intensity of the threat, the proximity of the threat, and also the context in which the threat is occurring. And behaviors such as flight or defensive attack are believed to be higher up the scale than behaviors such as freezing. So why would one want to understand the circuits that mediate transitions between these states? Well, perhaps understanding how these circuits operate in the healthy brain will help us understand what goes wrong in exaggerated states seen in anxiety disorders such as PTSD, panic, and specific phobia. So the problem is that for many decades, we've used Pavlovian threat conditioning to understand how the brain mediates fear, learning, and memory. And that's fine, and we've learned a tremendous great deal about it. What I'm illustrating here is a typical auditory threat conditioning paradigm. So animals are conditioned with pairings of an auditory tone with an electrical foot shock within this context. And then 24 hours later, you can assay two different types of learning. You can assay learning to the context by placing the animal back into the context, and you can assay learning to the cues by placing them into a neutral environment, playing the cues, and looking at the behavior. What I'd like to illustrate is that almost exclusively what's reported in the rodent fear literature are freezing values. Now, what you saw in that video was that mice and all organisms have a complete set of defensive behaviors. And understanding the richness of these behaviors and how the brain generates them is tremendously important. So, at the beginning of my postdoc, I set out to try to understand whether we could use simple conditioning procedures to elicit these higher intensity defensive states. So I made several modifications to traditional Pavlovian threat conditioning paradigm, such as increasing the threat intensity by increasing the magnitude of the foot shock, increasing the number of pairings of electrical foot shock, and in fact doing multiple days of conditioning, and also testing behavioral responses within the conditioning context to see if responses to the auditory stimuli would be different within the conditioning context. So I don't have time to tell you all the experiments that led to this point, but in the end, what we developed is called the conditioned flight paradigm. And in this paradigm, we use what's called a serial compound stimulus, which is a 10-second tone followed by a 10-second white noise, which is paired with a strong electrical foot shock during conditioning. Now, what I'll show you in this video is what the behavior looks like as a product of conditioning. Now, hopefully what you could see there was that the behavioral response to those two components of the serial compound stimulus were quite different. And the bottom line is that what you see during that white noise period is profound flight, indicated by the escape jumping attempts and forward locomotor response, whereas during the tone component, freezing dominates. So it's possible to induce both passive freezing and active flight responses using simple Pavlovian conditioning procedures. So I wanted to test whether this phenomenon was consistent with the threat imminence theory. So I did two experiments to test that. I took fully conditioned animals in this paradigm, and I did simple extinction learning. So I placed them back into the environment and played the cues over and over again without there ever being a foot shock outcome. And what we observed in black are the flight scores, in gray are freezing values. What is observed is that flight is rapidly diminished during extinction and is subsequently replaced by freezing. And this switch in defensive responses during extinction is consistent with there actually being a hierarchy of defensive behaviors, which this paradigm can assay. We also wanted to test whether this behavior was context-specific. So to test that, here that is the experiment where the animal's placed back into the conditioning context, and you can see that white noise elicits very high levels of flight. If you take the same animals and place them into a neutral context that they've never experienced before, you can see that there's absolutely no flight that is observed there. What does dominate in that environment is freezing responses. So what you can see is that in the neutral context, there are escalating levels of freezing to those components of the serial compound stimulus when they're played in the neutral context. And so what this illustrates is that white noise elicits more intense defensive responding in both contexts, culminating in flight in the conditioning context and significantly higher freezing in the neutral context. So to summarize this, the conditioned flight paradigm allows for rapid stimulus-specific behavioral switching. It's context-specific, and the extinction experiment shows that there is a hierarchical organization of defensive behavior. And we believe this is a good model system to start studying defensive response intensity. So the question is, where should we begin this investigation? So where should we look to understand how sensory input is processed by neural circuits to ultimately induce what can be very divergent behavioral outputs? So our first candidate structure was the central nucleus of the amygdala, which for decades has been known to be a main output nucleus of the amygdala that mediates defensive responding to threat, especially conditioned threat. So the central nucleus is a GABAergic structure. It's kind of like a little mini copy of the striatum. And it sends projections to neuromodulatory systems, hypothalamic regions, and midbrain regions such as the periaqueductal gray that are known to mediate autonomic and behavioral responses to threat. Now, very careful neural circuit dissection has been done in the past, and what these studies have illustrated is that there exist different inhibitory motifs within the central amygdala that act to control conditioned freezing responses. And so what I'm illustrating is some of those findings here, where you have cells such as somatostatin-expressing neurons and other neurons expressing PKC delta, which are engaged in an inhibitory battle to determine whether the output coming from the central amygdala to the PAG will be activated or not. And when that output is activated, you will elicit a freezing response via the PAG. But there also exists, in addition to these somatostatin, which promote freezing, and PKC delta cells, which inhibit freezing, there's another population of cells called, that express corticotropin releasing factor. And nothing was basically known about how these neurons mediate defensive behavior. So we asked the question, are these central amygdala freezing circuits involved in conditioned flight? And just as is true for freezing, can inhibitory interactions be important for the selection of defensive responses? So the first thing we did is we used optogenetics to inhibit these populations in the conditioned flight paradigm. So the method is we use what are called Cree driver lines, which allow us to get expression from different viral vectors into particular types of neurons. We implant those virus-injected animals with fibers to target the light to the parts of the brain that we want to target. And we use an internally controlled design where we have half of the trials in which the light will be turned on during the white noise and half of the trials in which the light will not be turned on. And remember, the white noise is the period which elicits very strong conditioned flight responses. So what we saw was that if we inhibited PKC delta or somatostatin cells, there was no change in the flight response, which here is indicated by speed. And however, when we inhibited, using optogenetics, the CRF positive population, we were able to completely ablate the conditioned flight response. So this indicates that this population in the central amygdala is necessary for the conditioned flight response. So we also wanted to know at the neural level whether there would be neural correlates that would follow this behavioral finding. So for this, we made extracellular recordings of identified CRF neurons. These neurons are identified using optogenetics and a technique that's called phototagging. And what we found was that so shown here is normalized neuronal activity of CRF cells in the conditioned flight paradigm. And please note that there is a significant activation of these neurons to the white noise in the conditioning context. If those same animals are placed in the neutral context where freezing dominates, that excitation is gone. We also used phototagging to record from somatostatin positive neurons. And what we found there is that that population was inhibited to the white noise when the white noise was eliciting flight. However, those same neurons were excited during white noise when it was eliciting a freezing response. So we wanted to go a step beyond and do an analysis to look at if these neural correlates correlated also with the behavioral state in addition to the sensory stimulus. So for this, we did a transformation looking at the neuronal activity within and outside different periods of behavior. And what we saw was that CRF neuronal activity correlates with flight, but not freezing. And somatostatin neuronal activity was inhibited during periods of flight and was excited during periods of freezing. So this begs the question, do these cell types interact? These reciprocal activity profiles might indicate that these cells interact. So to test this at the behavioral level, we used an optogenetic gain-of-function approach. So for this, we used excitatory optogenetic tools. We put channel rhodopsin into the CRF neuronal population, and this time we are exciting those neurons during the tone period to see if we can get the animal to bias away from passive freezing and towards active flight. And indeed, that's what we saw. We saw an increase in active behavior and a decrease in freezing behavior when we stimulated CRF neurons. With the somatostatin population, we wanted to stimulate them and try to bias the animal away from the active response and towards freezing. So we stimulated during the white noise, and what you can see is that there is a decrease in active behavior and an increase in freezing behavior. Okay, so we wanted to see what the putative mechanism of this might be. So for this, we moved to a brain slice preparation. I collaborated with a very talented in vitro physiologist named Sabina Krab. And what was determined from these experiments, I'm running out of time, so I'll just race through. What was determined from these experiments was that CRF cells preferentially provide GABAergic inhibitory input to somatostatin cells, which is strong enough to block current-evoked spiking in those neurons. And the reciprocal is also true. Somatostatin cells provide very strong GABAergic inhibition to the CRF neurons. And the bottom line is that this indicates that these central amygdala cellular substrates of flight and freezing have reciprocal inhibitory connections. So what's the bottom line? We believe that this provides a mechanism by which competitive inhibition could act to select different defensive behaviors in the central amygdala. And in the last 10 seconds, I'll just tell you the central amygdala doesn't sit alone. So we are now targeting the downstream outputs of the central amygdala, in particular in the PAG. And if I'm allowed to go over just 10 seconds, the reason we think that the PAG is a good target, because if you stimulate glutamatergic cells in the PAG, you get the following behavior. So stimulation in a certain column of the PAG of glutamatergic cells will induce very robust flight responses. And the central amygdala, we've determined that the CRF neurons, which promote flight, project to the PAG, and so we believe that this might be the pathway that mediates conditioned flight. And similarly, we've used a monosynaptic rabies-based approach to start to identify the putative glutamatergic inputs to the CRF population to try to target and figure out where the excitation source is that's exciting these GABAergic neurons. And with that, I will just acknowledge my lab and my current and past funding sources, and I'd like to thank you for your attention.